My name is Juliana and I spent this summer with at the Naval Medical Research Center with the Undersea Medicine Department. My project was focused on the effects of oxyhemoglobin affinity on high-altitude cerebral edema. Now, our project, in the context of the military, is meant to help service members that are stationed at high altitude, such as mountainous regions in Afghanistan, who are not given proper time to acclimate to the hyperbaric hypoxic conditions that they are exposed to. This puts them at risk to a spectrum of disorders, starting with acute mountain sickness and eventually progressing to high-altitude cerebral edema. Now, high-altitude cerebral edema, or HACE, is characterized by increased cerebral vascular permeability in the brain, causing leakages to the surrounding tissues and swelling in the brain. Now, we are testing a compound called 5-HMF that is meant to help alleviate the effects of high-altitude cerebral edema by causing a left shift of the oxyhemoglobin dissociation curve. This would increase the affinity of hemoglobin to oxygen in the blood and help support O2 loading into the surrounding tissues to help prevent the vasculature from leaking fluids. Now, in terms of the military, this would help the servicemen from decrements in cognitive performance and also a difficulty in operational functions due to the effects of high-altitude cerebral edema. Now, the three things I would like to highlight on this poster are the HACE model that was implemented in the study, the effects of P50 values that we got from using 5-HMF, and also the dosing study that we designed in response to our results in the study. So, with our HACE model, it was implemented in the study and developed by our lab previously, and we flew rats in the hyperbaric chamber, as seen here, at 24,000 feet for 24 hours to simulate high-altitude cerebral edema. Now, in this study, we had a control group of 9 rats that received saline and an intervention group of 7 rats that received 5-HMF. So after the rats were flown, 30 minutes later, blood samples were collected and sodium fluorescein was injected into a tail vein. Now, the sodium fluorescein was used as a way to measure the edema in the brain, as after the necropsy of the animal, we harvested the right hemisphere of the brain and measured fluorescence in the brain tissues to see if any sodium fluorescein leaked from the vessels into the tissue to measure edema in the simulated altitude. Now, afterwards, the blood samples that were taken from the animals were used in order to study P50. Now, P50 is a point on the oxygen hemoglobin dissociation curve where 50% of hemoglobin is bound to oxygen. Now, this is a point that's used to measure points where oxygen is being used in the body, and at hyperbaric hypoxic temperatures, a lower partial pressure of oxygen will often mean that tissues are being deprived. Now, 5-HMF is expected to cause a left shift of this, and we expected to see this as well in our animals. In this study, we did not get the same results, and as you can see in this figure here, on the x-axis we have our treatment groups, and on the y-axis we have our hemoglobin saturation percentage. The means of the two groups are graphed here, and they were found to have no significant difference between the two groups. Now, in response to this, we decided to investigate several factors that could have led to 5-HMF being ineffective in this study. We chose to focus on the dosing aspect of the study, and as you can see here in figure 4, designed a timeline of a dosing study experiment that we can use to help test which concentrations of 5-HMF would be most effective for the study. Now, here you can see we'll be taking P50s 10 minutes before a dose of either 40 mg per kilogram or 100 mg per kilogram via an intraperitoneal injection of 5-HMF. P50 values will then be taken 2, 4, and 8 hours prior to the dose. Now, the reason why we chose 40 mg and 100 mg per kilogram are due to previous literature studies that have found success with these concentrations in other animals or also rats as well. In our study, we used 20 mg per kilogram, and they are administered first via a bolus 1 milliliter injection into the animal, followed by a subcutaneous osmotic pump that was kept in the animal throughout the duration of their flight as a maintenance dosing of 5-HMF. So in this study, we decided to test 40 and 100 mg per kilogram, and in figure 5, you can see some preliminary results that we found from the study. The green lines show the P50 shifts of animals that have had 40 mg per kilogram, and the black one shows animals that have had 100 mg per kilogram. As you can see so far, the black seems to have caused a greater left shift in P50, indicating that this may be the proper dose to be used for studies of this nature, but we will continue to follow through with this study and hope to optimize the concentration of 5-HMF that we can be using in these types of studies in our lab. Now, in conclusion, basically our HASTE model was effective in this study, and the effects that we got for P50 after using 5-HMF, while unexpected, led us to further investigate the factors that could have led to that and design a dosing study to help us optimize the concentration of 5-HMF that can be used in the future. Now, our lab will continue to do acute hypobaric hypoxic experiments using 5-HMF, as well as some chronic experiments where research like this, determining which concentration will be most effective, will be especially important to maintain those effects of 5-HMF and continue the research that we are looking to do in this area.